devote the next 2 sessions to methodologies that are commonly used in cancer research, because if you go to seminars or if you read the cancer literature, you're obviously going to see a lot of data slides, and I think that it's important for you to understand the methodologies that are used to generate this data. So surely what you've picked up by now is that cancer is characterized by numerous genomic alterations, loss of chromosomes, gains of chromosomes, chromosomes breaking and recombining, deletions of little parts of the chromosomes, amplifications of parts of the chromosomes, and there are also what are called epigenetic changes, which are changes that affect gene expression but don't affect the sequence. They can affect, for example, DNA methylation, which Dr. Soares, I think, is going to discuss. Dr. Soares is a professor of pediatrics at Northwestern Feinberg School of Medicine, and he is also director of cancer biology and epigenomics. This is a program at the Children's Memorial Research Center. So he's going to discuss basically methodologies used to detect alterations in the DNA, both at the genetic level and at the epigenetic level.
Thank you. Just begin by saying that it's a real pleasure for me to speak to you today. One of my best friends, John Quackenbush, who is a brilliant biologist, he's a professor at Harvard. He started as a physicist. So usually it's way easier to go this route than the other. You guys are way smarter. So so it's a privilege to be talking to you today. I try to make this broad enough to raise interest. I take that this is an opportunity just to get a flavor so that you develop interest, and I will be glad to discuss further with you, and you can contact me afterwards too. So I'm going to cover these topics. And you're going to get sick of me today because in the afternoon I have another talk, which is more on epigenetics and genetic variation and epigenetics. But now I'm going to cover a little bit of cloning, PCR, how we introduce DNA or RNA into cells, a little bit of a discussion of the central dogma that's no longer from genes to proteins, how our genomes codes for proteins, and a little bit about the Genome Project and the impact that the Genome Project had on cancer research. And I'll end with a little bit of normal and disease variation, which is an introduction for this afternoon's session. So let's talk about cloning. So cloning is a way to amplify a specific gene or a fragment of DNA that you want to investigate or use to test function, for example. So there are these circular DNA molecules, plasmids, which have sequences that enable them to replicate inside the bacteria. So what one can do is, let's say that you want to clone a human gene. So you want to make multiple copies, have massive amounts of that human gene to do analysis with. So you can you take a genomic DNA, you extract that genomic DNA, and you do a digestion with a restriction endonuclease, which is a type of enzyme that recognizes a specific sequence. In this case, EcoR1 is GAATTC, on the DNA, and wherever that GAATTC will occur, it will clip, but it clips in a way as to leave an overhang, a sticky end, such that if you do the same with the plasmid, now you have two molecules that have two types of molecules, both of which have the same sticky ends, so they are complementary. So you can do in vitro a ligation as to insert these sticky ends of this fragment into the plasmid, and you do the ligation, you make a circle again, and now you transfect this DNA into bacteria. You grow the bacteria, and then you get amplification of your clone. So how come you don't get overgrowth of bacteria that didn't uptake any of the plasmid that you meant to introduce into the bacteria? Because this plasmid, in addition to having a replication, a sequence that enables replication, also has a selective marker, for example, an antibiotic resistance gene, so that if you grow the bacterial culture in the presence of that antibiotics, only the bacteria that uptook that plasmid will grow. So when you plate, you get colonies that have the foreign DNA that you introduced. So often enough, you have minuscule amounts of DNA. And you need to amplify that DNA before you can clone it. Or you don't need to clone it, because if you have the ability to amplify, you may not even need to clone. You may get enough to do what you need to do. Sometimes you have to clone to put in vectors that will enable functional analysis inside the cells. But the PCR, polymerase chain reaction, is extremely powerful. And let's see if this works. It's
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. Chromosomes are not always present. They form around the time cells divide, when the 2 copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
Okay. So you can see how powerful it is. You can go from a single molecule to a billion molecules in 30 cycles or something. So it's really powerful. So now you know how to clone DNA, and you know how to do PCR. So now, what if you want to introduce DNA into cells to do functional analysis? So there are different ways to do that. I will give you examples of some of the most used methods. So the process of introducing DNA into cells. It's called transfection. So it's the process whereby you take nucleic acid sequence, DNA or RNA, and introduce either biochemically or by physical processes. I will give you examples of each. So this is one method using Dextran. Dextran facilitates the binding of DNA to cell membranes. So what happens is that there is endocytosis. The DNA that is complexed with the Dextran will be engulfed by the cellular membrane. It's a process of endocytosis. Then you get these vesicles, and then that way you have the DNA into the cell. It doesn't get integrated in the chromosome. It stays as an extra chromosomal fragment. The second method, which is the most widely used, is electroporation. It uses high voltage to introduce DNA into cells. What the high voltage causes is the formation of pores in the cell membranes through which DNA enters. Then you harvest the cells and you plate and you get your cells with the transfected DNA. The other method uses liposomes. So these are cationic lipids. The chemical and physical similarities that they have to the biological, to the lipids in the cellular membranes is key to the process. There is this spontaneous formation of complexes with DNA. It's called lipoplexy. So it's these lipids complex with DNA, and then with that, they get into the cells. You can also use virus to insert DNA into cells in processes that are called transduction. Slightly different methods are used for transfection into mammalian cells, but again, electroporation is a very common one, and the use of calcium phosphate based technologies is also quite used to be the most popular, but it's still used in some case. But electroporation is the most efficient way to do this. I'm not going to cover microarrays much because that's going to be talked in the next session. I just want to make the point that it's possible to do in mass transfection of DNA into cells. You can array DNA or RNA, and then you coat that with cells, and then you do the process that will make it possible for the DNA to get into the cells. So this is important for screening. Each well may have an RNA that specifically inhibits a gene. So you expect a certain phenotype when that gene is inhibited. So if you have a number of different RNA molecules here, only one of which will inhibit that gene, then only one of the wells will display that phenotype. So that's one way to do large-scale screening to find what is the specific RNA that will affect that gene, and what's the phenotype that you have when that gene is inactivated. That will be covered later. So, in conclusion, cell transfection is widely used. There are many applications. You can produce protein because the DNA, the gene that you put in bacteria, under proper conditions, that can be done so that the bacterial culture, that plasmid will produce the protein that's encoded by the gene that you put in the bacteria. And then you can make massive amounts of that protein and use pharmaceutically or for whatever the application may be. Changing the protein expression profile of a cell to assay for the effects of a gene. So you can overexpress a gene and see what's the consequence to the cell. Alternatively, you can use these RNA interference based approaches to inhibit the expression of a gene and see what the phenotype of the cell is upon inhibition of that gene. And you can add genetic markers to a cell line. So there's a number of applications that with transfection you can do. So we used to think of our genome as encoding proteins, and that proteins were the main and only molecules that were responsible for the function of our cells. Here we learned a lot with the Genome Project about that, and this has been, this dogma has changed quite significantly, and we'll discuss that. So from DNA to protein, we first have to do transcription to copy DNA into RNA, and the RNA in the cytoplasm will be translated to protein. So let's see how transcription works.
What you are about to see is DNA's most extraordinary secret, how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
So then the RNA goes to the cytoplasm and it's translated to proteins.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
So, this is the last one I want you to see.
Think of how many letters fit on a single page. And now imagine a stack of pages 300 feet tall. That's how much information is stored in the DNA inside every human cell, the entire human genome. If you sort through the 3 billion letters that make up the human genome, you find some surprising things. Only about 1% of the 3 billion letters directly codes for proteins. Of the rest, about 25% make up genes and their regulatory elements. The function of the remaining letters is still unclear. Some of it may be redundant information left over from our evolutionary past.
So the point that I want you to take home is that we used to think that the genome was composed of DNA genes that code for proteins and junk sequence, mostly repeats, sequence that were present thousands of times in the genome, and that they were just junk. With the Genome Project, with all the sequencing effort, it started to become pretty clear that the component of the genome that actually encodes protein, like you saw in this, it's only 1%. The vast majority doesn't code for any protein. And what we used to think that was junk, these repeats, have now started to be better understood, and they are now thought to play important roles in the organization of chromosomes, in the stability of chromosomes, in the epigenomic. I'll talk about these modifications that occur in DNA that are not encoded but are important for the function, which is the epigenetic code. So all these sequences that were thought to be junk, most of them are probably not junk. But most importantly, there are many, many non-coding RNAs, sequences that are not repetitive. Simply they don't code for any proteins, but they play a major role in the control of gene expression. Some of these, for example, microRNAs, tiny RNA molecules that actually can, they are complementary to sequences in mRNAs derived from genes, and when they anneal to those sequences, they can either cause degradation of that RNA or inhibition of translation of that messenger RNA. The outcome being no protein from that mRNA being made. So these non-coding RNAs, they are very important in controlling cellular processes. So we have come a long way, and we have now a whole new era that has emerged as a result of the Genome Project. And a number of technologies, I'm going to skip this because it's not loud enough for you. A lot of the technologies that were developed so that the Human Genome Project could be accomplished within a reasonable time frame, which required a lot of robotics, was developed very quickly thanks to the needs of the Genome Project. And these technologies, high throughput screenings and things like that, and most importantly, high throughput sequencing, that was a necessity because of the Human Genome Project. All these was developed, and I think we're getting closer and closer to the day when it will be possible for everyone's genome to be sequenced, and then we will understand better normal variation and the association of variation with disease. There is a continuum of genetic variation from single nucleotide changes called SNPs, single nucleotide polymorphisms. A polymorphism is when you have one variant that is present in at least 1% in the population. It's considered polymorphic. So you can have single base pair changes all the way to chromosomal variation. The genome has, our genomes differ in many ways, not only the presence of a single nucleotide polymorphism, but also chunks of DNA that are deleted or amplified. There are multiple insertions and deletions. Many of the repeats that I mentioned, they actually are mobile elements. They can by an RNA-mediated mechanism, they can be copied and pasted. A DNA copy of that particular repeat can be converted into RNA. That RNA can be inserted another place in the genome, and in the genome be converted back to DNA. So this is called a retrotransposable element. So these are all sources of genomic variation. We have large segmental duplications, big chunks of DNA that are present in more than one copy. So there's a tremendous amount of variation in our genomes, and there are methods that are used to ascertain that variation. You can take a reference genomic DNA and a test genomic DNA labeled fluorescently with different colors, and you can have an array on a chip that will have sequences representing each chromosomal region, each piece of DNA in a chromosome. And if the reference DNA and the test DNA, if each sequence were present at the same, with the same amount, you would have only yellow. But if you have a particular piece of DNA that's present in more copies in the reference DNA than in the test DNA, for example, then you're going to have, wherever that sequence is, you're going to have a red instead of a yellow, showing that there are more copies here than there. So this way you can ascertain copy number variation in a very high throughput manner. And these are it's just a depiction of the segmental duplications that I mentioned to you. About 5% of the genome contains these regions of segmental duplication. So these are chunks of DNA that are at least 90% identical and are larger than 1 kilobase. Some are much larger than 1 kilobase. What is important is that these large regions of duplication make recombination more likely to occur. So you may have mispairing of these regions, and then when you have recombination, you end up with these type of product that will have a duplication. You have two these, and you have a deletion. So a very important field that has emerged from the possibility to identify genetic variation in populations are the studies that attempt to associate variation with disease. And there are many cases where a particular variation has been associated with disease. And here are some examples of genes that when you have decreased copies, for example, you may cause an increased susceptibility for HIV AIDS, for example. There is also another type of variation that's way more rare, and it's not a normal variation when you have aneuploidy, which is when you have one chromosome, a difference in number of chromosomes. So the alteration is not just in copy number of a fragment of DNA, but a whole chromosome. Down syndrome, for example, is when you have 3 copies of chromosome 21. And here is a karyotype, a display of all the chromosomes. You remember that they are displayed in pairs because our genome is a diploid genome. We inherited one chromosome from each pair from our father and the other from our mother. So we have a diploid genome. So there are alterations, structural alterations that are microscopically visible with karyotyping. So this is an example. You can so-called painting, chromosome painting, with probes that cause each chromosome to light up with a different color. And then you can see, you can see, for example, translocation. This chromosome 7 has a piece from chromosome 13 and vice versa. So this is a case of balanced translocation. So all the sequences are there, but not where they're supposed to be. And this is a similar to what I talked before about the type of array hybridization that can be done to identify copy number alterations. You can do that arraying large fragments of DNA or oligonucleotides, and of course then the sensitivity is different. And these are just examples of how duplicated regions can make those sites more prone to undergo structural alterations by recombination. So, take one example like this one. This pair of homologous chromosomes are mispaired here, and then you end up with a deletion and a duplication after the crossover. So you have DCB and then goes to the other CBA. So you end up with a duplication of BC. That's, and then therefore the other product will have a deletion of BC. So I want to show some applications of the type of technology that we talked about, stuff that we did in the lab. This is work with neurospheres. So when you grow neural stem cells, you can grow under conditions that these neurospheres are formed. These neurospheres could be formed two ways. You could have a neurosphere that would be composed of the same kind of cells that duplicated to form a group, or like that, or you could have a mixture of the two. So the question is, when you have these neurospheres, you have these multiple, these conglomerates of cells, are they all derived from the same neurostem cell, or are they the result of the merging of different cells to form a bigger clump? So what we did was we used a viral vector that had a sequence that encodes this green protein and a red protein. You always have to have a drug resistance to do your selection. So what we did was we took a bunch of cells of these neurostem cells, and we transduced with the virus that produced that green protein. We took another batch of neurospheres and we transduced with the virus that made the red protein, and then we mix the two. And then the question is, when what you're going to see is single stem cells, when they form neurospheres, are you going to see all to be green and red, or you're going to see this kind of scenario? So if we see these, it's because we have the merging of different stem cells forming a neurosphere. If we see these, it's because each divides, and then the neurosphere is all derived from the same. And what we see is that you have these mixtures, so they result from the merging. Another example is the studies that we do to investigate behavior of tumor cells in vivo. So there is a possibility to take the firefly luciferase gene, that the firefly luciferase gene makes the cell luminesce. So if we take a viral DNA construct that has this gene in it, and we transduce that virus to a cell, now that cell will luminesce, and there is an instrument that can detect cells that are luminescent. So if we take a tumor cell and we introduce this virus with this firefly gene, luciferase gene, then we're going to be able, without having to sacrifice the animal, monitor progression of this tumor in vivo over time. And just to introduce the presentation that I will make this afternoon, just so that you realize how significant these epigenetic alterations that I will discuss with you later can be. In this case, we did an experiment where we took a particular type of cancer cells, in this case a cartilage tumor cell, and we exposed to a demethylating agent, an agent that will cause the DNA not to have the cytosines that are in CpG dinucleotides methylated. And that had a profound effect on the aggressiveness of the tumor cells. So the same amount of cells, when we had with and without the treatment, we were able to see a much larger tumors when the cells were treated with the methylating agent. And we even saw cases of metastasis in the lungs, which had never been seen with these cells. So this is the untreated and this is the treated, showing how much larger the tumors became when that particular DNA modification occurred. So I'll talk about these epigenetic mechanisms in the afternoon, but this is just an introduction so that you see how functionally significant they can be. So sorry the videos were not loud enough. I'll take questions. Yes.
I always wondered, in the standard transfection methods, I understand how the DNA, sorry. Anyway, I understand through common transfection methods how DNA enters across the plasma membrane, but do you have a sense for how the DNA actually ends up within the nucleus so that it has access to the gene expression machinery there?
Now that's, there's a lot, I mean, especially with electroporation, I think that there's a lot that's still a black box. Yes?
So in general, are point mutations or chromosome translocations and fusions, is one or the other more important in development of the genetic changes that lead to cancer, or? Oh.
Yeah, I mean, that's a great question, and I hope to cover that in more detail in the afternoon. But the chromosomal alterations are much rarer. The single nucleotide polymorphisms, because they are polymorphic, you know, as we're calling them polymorphic, they occur in normal population. So not necessarily presumably the majority is neutral, right? So it is probably the combination of these of occurrence of these variants that may be predisposing to a certain condition. But we will talk about that in the afternoon. Any more questions? Yes.
Given that most research seems to have been done on either cell lines or individuals who are in treatment, how well can we classify things that are normal variation versus things that are disease variation? Right. Since there's obviously a greater pool of research on people who are diseased rather than going around and collecting healthy individuals.
Right. Again, this is the topic of the afternoon session. But I mean, this is a great question, and you have to have well-defined groups of normal people and disease, and then you have to have detailed genotyping of the individuals in these two groups and do association studies to see if you find a certain overrepresentation of variants in the disease group. That's basically what is required.